Protists learners. My name is Wesi Musile. I am the subject advisor for life sciences from the Free State province in Muteo district. So today, our topic for today is biodiversity and classification of microorganisms. And then when you look at our ATP, so this topic is covered from week number 1 up to week number 3 of 2021. And then let us look at the first week. So under microorganisms, this is what we have to do. We have to know the basic structure and general characteristics of the following groups of microorganisms. Number 1 is viruses, bacteria, protists, and fungi. And then after that, the roles that these groups play in maintaining the balance in the environment and the web of life. And then from week 2, this is what you have to do. Symbiotic relationships of bacteria, such as nitrogen-fixing bacteria in plants and E. coli in the human intestine. And then after that, we will move to the effect and management of one disease from each of the 4 groups. And then under viruses, it's either we do rabies, HIV, AIDS, or influenza. But for today's lesson, we will focus on rabies. And then bacteria, and then the disease that are caused by bacteria, we have blight, cholera, tuberculosis, and anthrax. But for our topic, we will only do cholera. And then under protists or protista, we have malaria. And then fungi, we have rust, thrush, ringworm, atlas foot. So we will only do the ringworm. And then when we move to week number 3 of 2021, and then this is what we have to do. Under immunity, we will do immunity, including plants and animals' immune response against the infecting microorganisms. And then after that, we do vaccinations. And then we will also cover the use of drugs. For example, we have antibiotics and the effect on microorganisms. So meaning that the effect of antibiotics on microorganisms. And then we will also cover the use of microorganisms to produce medicines, such as, for example, insulin and other antibiotics. And then the last concept will be the traditional technology to produce, for example, beer, wine, and cheese. So before we start with this topic or chapter, so these are the terms that will be covered in this topic. And then the first one is microorganism, diversity, acellular, unicellular, prokaryote, eukaryote, parasite, pathogen, capsid, plasmid, autotrophic, heterotrophic, chemotrophic, saprophytic. And then we also have binary fusion. And then we will also cover sporangium, rhizoids, stolon, sporangiophore. And then sporangium, rhizoids, stolon, and sporangiophore, and spores. These are the parts of the bread mold. Normally they are found on the structure of the bread mold. And then we also have the antigen, antibody, antibiotics, immunity, vector vaccination, and genetic engineering. So I'm sure that you still remember this table from grade 10. Okay. And then this table or the flow diagram, it represents the 5 kingdoms of living organisms. We also did this in grade 10. And then we also remember that the 5 kingdoms of living organisms, and then we have Monera, and then an example, it is the bacteria. We also have the protista or protists, and then fungi, plantae, and animal. The last kingdom will be the animalia or animals. And then let us also look at this flow diagram. And then this diagram, it represents the characteristics that are used to classify organisms into these 5 kingdoms that we have mentioned. So When we start at this point, we have all living organisms. So it means that all living organisms are divided into prokaryotes and eukaryotes. And then what are prokaryotes? Prokaryotes, and then it means that are all the organisms that don't have a definite nucleus. And then another part, it is this one of the eukaryotes, and then all organisms which have definite nucleus. So organisms that fall under prokaryotes, we have the kingdom Monera. And then you also remember that I said an example of kingdom Monera, it is the bacteria. So kingdom Monera, they are prokaryotes because they don't have the nucleus. And then another one, let us move to this one, to the eukaryotes. So eukaryotes are divided into organisms which are unicellular and multicellular. So for unicellular, we have kingdom Protists. And then for multicellular, we have the plantae and kingdom Fungi. But still again, under multicellular is divided into two. We have organisms which are autotrophic and organisms which are heterotrophic. And then you still remember that autotrophs are all the organisms that can manufacture their own food. And then heterotrophs, they cannot manufacture their own food. So under autotrophic, we have kingdom plantae or plants. We know that all plants are able to manufacture their own food. And then under heterotrophic, and then this is where we have kingdom Fungi and kingdom Animalia. Both these kingdoms or groups, they fall under heterotrophics because they are not able to manufacture their own food. So again, we have under the heterotrophics or heterotrophs, they are divided into two. We have these organisms where these ones, they digest food and then takes it in. So all those organisms will be the fungi. But kingdom Animalia, or which are animals, they take in food and digest it inside the body. So that is all about all the 5 kingdoms that we have of the animals. And then again here we have a diagram. Here is a diagram here which shows the eukaryotes and prokaryotes. And then look at the eukaryotes. We have the plantae or plants, and then here is a diagram, and then we also remember that all the plants, they are multicellular. And then we also have fungi. It also falls under eukaryotes, and which is also multicellular. And then we have animals. Here is a butterfly, which is also multicellular. And then we also have the Protista or Protists. And then on this diagram, we see an example as amoeba, which is unicellular. So it means that it consists only of one cell. And then prokaryotes, we only have, you still remember that we said that under prokaryotes, we have the kingdom Monera. And then when you look at this diagram, this is the diagram of the bacteria. So bacteria is an example of Monera. And then let us continue with our lesson. So let us look at, all right. As you remember that I said the topic for today is microorganisms. So under microorganisms, we have the microscopic. We have those organisms that we refer them as microscopic, and then other organisms, they are microscopic. So if you remember that all organisms which are macroscopic, it means that are all these organisms which can be seen with the naked eye. Be seen. And then micro, they cannot be seen with the naked eye, and then it means that we have to use the microscope. for us to be able to see them. And then let me give you an example of macroscopic. Here we have what? The human egg cell. Example, it will be the human egg cell. And then another one, the human egg cell, you still remember that the size, it is very small, but at least we can be able to see it. And then all the human egg cell, they are the size of a dot. And then again, we have an ostrich eggs. So when we move to microscopic, like as I have said that microscopic, we can't see them with our naked eyes, and then we have to make use of the microscope. So we have bacteria here. Bacteria, they are very small. So they can only be seen with the use of the microscope. And then all the microorganisms, so all these organisms which can be used, seen by the use of the microscope, and then are called microorganisms. So, and then again, microorganisms, they are found in all the 5 kingdoms. All these 5 kingdoms that we started with, which are Monera, Protista, Plantae, Fungi, and animals. And then again, there is another microorganism that is also found in the 6th group, but it cannot be placed in any of the 5 kingdoms that we have mentioned. And then that microorganism are viruses, or it is a virus. So let us start with the first microorganism, which is a virus. So like as I have said that the virus, it is also found in the 6th group, in group number 6, but it cannot be placed in any of the 5 kingdoms which have been mentioned. All right. And then let us go back to our ATP. When you check our ATP, we said that from all these groups of organisms, what is it that we have to study? We have to study the basic structure and characteristics. So let us start with the general characteristics of viruses. So bullet number 1 here we have, they are microscopic. So it means that they are very small, and then they can only be seen with the use of the microscope. And then they are not cells, since they are without a cell membrane. So it means that they don't have the cell membrane and other components of the living cells. So that is why we say that they are not the cells. And then number 3, they contain a nucleic acid, DNA or RNA, and then which is surrounded by a protein coat. And then another general characteristic, they use the nucleic acid and ribosomes to reproduce. They remain inactive outside a living cell. So it means that they are non-living, or they become non-living when they are outside a living cell. But they replicate inside a living cell. So it means that they live inside a living cell, and then again they cause diseases. So it means that they are pathogens. You still remember that all organisms that cause diseases, we call them pathogens. And then again, viruses that attacks bacteria. We have those viruses that attack bacteria, and then we call them bacteriophages. page or bacteriopages. So those are the general characteristics of microorganisms, and then in this case we have viruses as examples. So let us move to the structure of a virus. So please remember that you also have to know the structure. So we have the head, and then inside here, this part of the head, it is where the DNA is located. We also have the neck, the collar, the sheath, and then the tail, fiber, and the base plate. So that is the diagram or structure of the virus. And then after that, we will move to the effects and management of rabies. So please remember that rabies is one of the diseases which is caused by virus. So you have to remember that for each and every microorganism, you have to know what is it that you have to know, the basic structure and general characteristics, and then after that you also have to know the effect and management of one of the diseases which will be caused by a specific microorganism. So for viruses, this is what we are going to study. We will study the rabies as one of the diseases which is caused by a virus. And then point number 1 for general knowledge. And then what is rabies? A rabies, it is caused by a virus which attacks domestic and wild animals such as dogs, cattle, etc. So rabies, it is a disease which attacks domestic and wild animals such as dogs and cattle and other animals that I won't mention here. And then this rabies again, it can be transmitted to humans by the bite of an infected animal. So it attacks domestic and wild animals such as dogs, and then it can also be transmitted by the bite of an infected animal. And then effects on humans. So under effects, this is what we will have. So a person infected with rabies will have to be incubated for a period of 1 to 60 days. And then another thing which is very important, and then what about the symptoms? Symptoms of rabies, they include headaches, and then fever, sore throat, nausea, diarrhea, and fatigue. So these are the symptoms of rabies. Headaches, fever, sore throat, nausea, diarrhea, and fatigue. And then again, a person who is infected with rabies may run about or may undergo convulsive seizures, and then again they can also produce large amounts of saliva. And then again they can also show a fear of water. Fear of water, which is normally called hydrophobia. And then again they can also have a difficulty in swallowing and breathing. So those are all the symptoms of rabies. And then how can rabies be managed? And then we will start first with people who work at animal welfare organizations. So it means that they will have to be immunized. All the people who work at welfare organizations or animal welfare organizations, so it means that they will all have to be immunized by taking a vaccine. A vaccine against rabies virus. And then another thing which is very important, they should also wear protective clothing. And then including gloves. I will just write gloves when working with these animals. So that is all about rabies. And then before we move to another microorganism, which is bacteria, I have one of the questions here, which is based on viruses or okay, so this is what we have. Read the following passage regarding the Zika virus. So this is another type of virus here. Zika virus, so we have an extract here, and then after that we have only one question, which is based on this extract. So Zika virus disease is caused by a virus transmitted by Aedes mosquitoes. People with Zika virus disease, usually they have symptoms that can include mild fever. So this is one of the symptoms. And then skin rashes, and then infection of the eye, muscle and joint pain, and headaches. So those are the symptoms of this virus, which is called a Zika. These symptoms normally last for 2 to 7 days. There is no specific treatment or vaccine currently available. The best form of prevention is protection against mosquito bites. The virus is known to circulate in Africa, America, Asia, and the Pacific. Recently in Brazil, local health authorities have observed an increase in Guillain-Barré syndrome, which it has to go in hand with Zika virus infections in the general public, as well as an increase in babies born with microcephaly, which is the abnormal smallness of the head in northeast of Brazil. So this is our extract, which is based on one of the viruses, which is called Zika. So here we move to a question. We only have one question here. Question 2.1.1, and then it says, according to the paragraph, the symptoms of this disease are not severe. And then let us go back to our paragraph or extract. So it has been said that the symptoms are not severe. Here are the symptoms. We said that it is mild fever, skin rashes, and then infection of the eye, muscle and joint pain, and headache. So our question goes like this. Why does the WHO, we still remember that it is the World Health Organization, want to prevent this disease. So it means that the symptoms are not severe, but still who wants to prevent this disease. And when we go back to our extract, let us check from this line. Recently in Brazil, local health authorities have observed an increase in Guillain-Barré syndrome. So it means that organization, which is the World Health Organization, they want to prevent this disease because of what? Because of an increase, because this disease or virus, it causes an increase in the incidence of Guillain-Barré syndrome. And then another thing, it is because it also causes an increase in the amount of babies born with microcephaly. And then we remember that microcephaly, it is the abnormal smallness of the head. So grade 11 learners, most of the time, questions which are based on this diseases which are caused by different microorganisms, most of the time you will get an extract or a background information, and then after that all questions will be based on that extract passage or background information. So this is one of the examples of the questions based on such diseases. So after that, we can move to the second microorganism, which is bacteria. So it means that now we are done with viruses, and then we move to bacteria or bacterias. And then again with the bacteria, we have to study the general structure and then general characteristics and the effect of one of the diseases caused by bacteria. And then most of the time, bacteria, they are about 50 times larger than viruses. 50 times larger than viruses, but still they are the smallest known living organisms. So you remember that with viruses, we have said that they have characteristics of both living and non-living organisms. So bacteria, they are living, and then they are still regarded as the smallest known living organisms. So bacteria, they are found everywhere in the air, soil, and water. So let us look at the general characteristics of bacteria. So here are they. They are microscopic, so it means that we can't see them with our naked eye. So we have to use or make use of the microscope for us to be able to see them. And then unicellular, it is only one cell. They are prokaryotes and have no organelles and no nucleus. You still remember that we said that prokaryotes are all the organisms without definite nucleus. Let me go back. Okay. And then they are capable of carrying out functions like nutrition, reproduction, excretion, respiration, and movement. And then they reproduce by forming spores or by binary fusion. So those are the general characteristics of the bacteria. And then let us look at the structure of a bacteria. So please remember that you also have to know this structure. So here is the structure of the bacteria. So we have the following labels: the cytoplasm and then the flagellum, which helps with movement. And then we also have the nucleus, the cell wall, and the slime capsule. So this is the structure or the diagram of the bacteria. And then now we move to the diseases. And then I also have this structure of the bacteria. We have the ribosomes here, the cytoplasm, cell membrane, again the cell wall, slime capsule, and the flagellum, which helps with movement, and inside we have the nucleus. So now let us move to, before we move to the disease, let us start first with some of the general characteristics which are not mentioned on the previous page. So, but with bacteria, some are autotrophic. We have bacteria which are autotrophic, and then others are heterotrophic. All right. So remember that autotrophic organisms or autotrophs are all organisms that are able, they are able to manufacture their own food. And then heterotrophs or heterotrophic organisms, they cannot manufacture. So they cannot manufacture their own food. And then we also have examples of heterotrophic organisms. So the first one, it is parasitic. So parasitic bacteria is an example of heterotrophic organisms. And then we also have saprotrophic bacteria. And then the last one is mutualistic bacteria. So parasitic bacteria, they obtain their food from other living organisms. So it means that they are parasites. They obtain food from other living organisms, and then it means that they are parasites. And then saprotrophic. And then before we move to the second type, the saprotrophic, and then again parasitic bacteria, they cause diseases. They cause diseases. So it means that they are pathogens. So you still remember that all organisms that cause diseases, we call them pathogens. And then when we move to saprotrophic, they obtain their own food from dead plants and animals. Dead plants and animals. So these are the saprotrophic bacteria. So they obtain their own food from dead plants and animals. So it means that they are decomposers. And then the last type, which is mutualistic bacteria. These ones, they live in or on plants and animals. For the benefit of both organisms. All right. And then let us look at effects and management of one of the diseases which is caused by bacteria. And then in this case, we are going to take cholera. So cholera is caused by a bacteria which is called Vibrio cholerae. So this means that when water or food contaminated with feces is swallowed, so cholera, it means that, or it simply means that when water or food contaminated by feces is swallowed, or that is the meaning of Vibrio cholerae. And then it affects the alimentary canal, causing severe diarrhea. And then it is also linked to insufficient access to safe water and proper sanitation. So that is all about cholera. And then again, when we look at this disease, it has been said that 75% of people infected with cholera, they don't develop any symptoms. 75% don't develop any symptoms, but this cholera, it stays in their feces for 7 to 14 days. So it stays in feces for 7 to 14 days. And then it also means that if a person does not wash his or her hands, so by this we mean that an infected person or a person who has cholera, if they don't wash their hands thoroughly and after that they touch food, so this bacteria, it may infect other people. So it means that always we have to wash our hands before, especially before we touch food. And then also, if there is no proper sanitation, the bacteria may pass back into the environment and then after that possibly infecting other individuals. And then again, 25% of people infected, and then they show a sudden onset of acute watery diarrhea, and then that can lead to death by severe dehydration. And then another thing which is very important, individuals with lower immunity, such as malnourished children or people living with HIV. Okay, malnourished children or people living with HIV, they are at a greater risk of death if they are infected with cholera. And then another thing which is very important, if this disease is left untreated, and then it can also kill healthy adults within few hours. So that is the effects of cholera. And then how can we prevent cholera? So the first thing, it is health education. So health education, so including the need to wash our hands with soap. So it means that we need to wash our hands, especially with soap, after defecation and before handling food or eating and drinking. So that is number one. And then number two, clean water and proper sanitation. For all the sectors of the population. All right. And then let us move to page number 5 to 6 of our question document. To wrap up bacteria. So here is our question. A grade 11 learner investigated the growth rate of bacteria. Bacteria can reproduce by binary fusion, which is by mitosis, in which a single bacterium divides into two identical offsprings. The table below shows the number of bacteria growing in a nutrient substance at 30 degrees Celsius over a period of time. So here is our table, and then on the first column we have time in hours, and then the second column, it is the number of bacteria, and then the units, thousands per millimeter square. So question 2.1.1, it says identify for this investigation the dependent variable. And then if you look at our table here, it means that the dependent variable, it will be the number of bacteria. So it means that the number of bacteria, they depend on what? On time. So the independent, by saying so, it will be time. Number of bacteria. So please remember that, learners, there should be that relationship between your dependent and independent variable. You can't say time depends on what? On the number of bacteria. But the number of bacteria, look at these numbers. We have 20 here, 60, 100, 1.50, and then they depend on what? On the time. So the second question, it says draw a line graph to show the change in bacterial numbers from 5 hours to 25 hours. So it means that this is the only information that you will have to use. From 5 to 25, it is up to here. So your graph will only be based on this information. And then let us look at our graph. This is how we have to draw our graph. So, and then another thing, please remember that always when you draw your graph, you have to include the caption. You start first with the caption, and then after that, the labeling of the X and the Y. For this graph, we have the time, and then please also remember to include the units. Here we have hours, time in hours. This is from our table. And then the labeling of the Y, we have our what? Our dependent variable here, the number of bacteria. So you still remember that. Another thing that you can do from our question, most of the time, when you formulate or you identify the dependent and independent variables, we always advise that you check on the what? On the first column. The first column, always it represents the independent variable, and then the second column, it represents the dependent variable. Or another thing that you can do, the dependent and independent variables, you can also get them from the investigative statement. So you will label your graph. We have our caption, and then labeling of the Y and labeling of the X. And then here is our information. Okay, the independent, the hours, 5, 10, 15, 20, 25. And then with the dependent, please make sure that your scale is correct. You have to use the correct scale. And then after that, you will plot your points from the table. And then another thing which is very important, learners, the distance between here, between the first value and the second value to the last value. This distance, it has to be the same, or otherwise you will be penalized for this. And then we can also check how these graphs are being marked. For writing the caption, and then for this case, the growth rate of bacteria between 5 hours and 25 hours. And then for writing the caption, you only get 1 mark. And then correct type of graph. You remember that our question, from our question, you were asked or required to draw a line graph. So if you have drawn the line graph, you will get that 1 mark. But for example, let us say you draw a bar graph or histogram instead of the line graph. So it means that you will forfeit that 1 mark of the correct type of graph. And then another thing, it is the x-axis from this criteria. So it says correct label for x-axis, appropriate scale, use, and unit. So it means that all these three things, they share this one mark. For writing the correct scale, correct label of the x-axis, which is time in hours, appropriate scale 5, 10, 15, 20, 25, and the units are hours. For all these three things, and then you will only get one mark. And then the same with the y-axis, correct labeling of the y-axis, number of bacteria on this graph, and then together with the what, with the measurements or the units, and then correct scale again and the units. Okay, you will get only one mark. And then for plotting, 2 marks only if all the 5 points are correctly plotted, and then you will get only 1 mark if only 1 to 4 points are correctly plotted. So please note again, this is also important. If the wrong type of a graph is drawn, like as I have said that you have been asked here to draw a line graph, and then you decide to draw another type of graph, maybe a histogram or bar graph. So it means that marks will be lost for correct type of graph, and marks will also be lost for joining of points. So it means that you lose 2 marks for correct type of graph and for plotting of the points or joining of them. And then if axes are transposed, so it means that, let us say for instance, instead of putting time here and the number of bacteria on the y-axis, so you take the number of the bacteria and then you put it on the x-axis, and then time in hours, you take it and you put it on the y-axis. So it means that the marks will be lost for labeling of the x-axis and the y-axis, because it means that the labeling of the x and the labeling of the y will be incorrect. So you will only lose 1 mark for that. So we are done with a question or an example of a question which was based on bacteria. So we now move to the third microorganism, which is the Protista or Protist. All right. So the Protists, they have the same basic, they don't have the same basic structure. Let me show you examples here of the Protists. Here are the examples. They don't have the same basic structure like with the virus and the bacteria. So they show a great deal of diversity in their structures. So here we have examples of the Protist or Protista. And then this one, it is a seaweed. And then we also have the dog vomit, which is also called the slime mold. And then after that, we have a variety or range of diatoms. And then we also have the green algae, which is the Spirogyra. And then again, another one, it is a range of dinoflagellates. So all these organisms, they are Protista or Protists. So let us look at the general characteristics of Protists. The first one, it says they are eukaryotic organisms. So it means that they are eukaryotes. And then you remember that we have said that eukaryotes are all organisms with definite nucleus. They have the nucleus. And then they are also unicellular. They are not multicellular like kingdom Animalia or animals. And others are multicellular. Okay, with Protists, there are those Protists which are unicellular, and then others are multicellular. And then they reproduce asexually by binary fission. and sexually by forming the spores. So we have two types of reproduction here. It is asexual reproduction by binary fusion and sexual reproduction by forming of the spores. And then some are animal-like, while others are plant-like. Like if you, as you have seen from the examples that I just showed here. When you look at these diagrams here, some they look like plants, and then look at the seaweed, and then also look at these ones, the diatoms, and the green algae, and then you also look at this one, the dinoflagellates, and then the dog vomit or slime mold. So the protists, there are those protists which look like plants, and then others look like animals. And then plant-like organisms or protists are autotrophic. So it means that all those protists which look like plants, they are autotrophs or autotrophic. So it means that they are able to manufacture their own food. So those are the general characteristics of the protists. So now we will move to one of the disease which is caused by the protists or protista. So we have malaria. We have malaria as an example. And then when we look at malaria, we know that it is caused by a parasitic protozoan, which is called Plasmodium. So malaria is caused by a protist or a parasite which is called Plasmodium. So again, malaria, it is spread by 4 different species of a group of mosquitoes that are called Anopheles. So normally this is what happens during an infection. I'm just going to show you this paragraph from this document. So this is what will happen. These mosquitoes, as I have said, that it is spread by 4 different species or a group of mosquitoes which are known as Anopheles. So these mosquitoes, this is what they do. They bite humans and then after that suck blood from them for their food. So it simply means that these mosquitoes, they depend on human blood for food. So they will bite and then after that they will also suck the blood. If one of them bites a human who is already infected with the malarial parasite or germ, which is called the Plasmodium. So the microscopic parasites are taken in with the blood. So it means that when they bite, and then what will happen, those microscopic parasites or the parasite will be taken in with the blood. So when this mosquito bites another human, so it will inject its own saliva into this person, and then to prevent the blood from clotting while it is being sucked up. It injects the saliva into the second person or another person. By so doing, it only prevents the blood from clotting while it is being sucked or while it is sucking the blood. So the malarial parasite in the mosquito or saliva enters the blood of the second human. So in this way, these mosquitoes, they spread the disease from one human to another. So this is how malaria is being spread. So then when we look at the effects or the symptoms of malaria, we have headache, and then fever, sweating, chills, muscular pains, back, which will be on the back and limbs, abdominal pains, diarrhea, nausea and vomiting, loss of appetite, and cough. So those are the symptoms of malaria. Or a person who is infected with malaria will show these symptoms. And then let us look at management of malaria. How can we prevent malaria? We can only prevent malaria by getting rid of the mosquito, by stopping the mosquitoes from biting us, and then also by taking medicines before entering a high-risk area to prevent the disease from developing. So this is how we can manage malaria. So before we move to fungi, another group which is called fungi, and then let us move to our question and answer document. Okay, so this question is based on malaria. It is on page 12 and 13, okay, of our document. So it goes like this. Malaria is a disease caused by a protozoan called Plasmodium. It lives in the blood and liver of an infected person. Malaria can kill, but more often it causes the victim to suffer from high fever, vomiting, and severe headaches. So this simply means that these are the symptoms of malaria which have been provided from this extract. So study the diagram below that shows the life cycle of Plasmodium and answer the questions that follow. So here is our diagram of the life cycle. All right. So we have this is the human being and then the liver here. And then step number 1, this is what happens. We have the mosquito here. So step number 1, the mosquito feeds on infected blood. So it means that the mosquito will bite a person, and then after that it will feed on the infected blood. And then number 2, fertilization in the mosquito's stomach. So after that, fertilization inside the mosquito stomach will take place. And then number 3, parasites produces thousands of immature offspring. And then after fertilization, and then immature offsprings will be produced. And then after that, parasites invade mosquitoes' salivary glands. So it means that these parasites, now they will move or they will invade or enter in the mosquito salivary glands, and then an infected mosquito takes blood or takes a blood meal, and then parasites enters the bloodstream and travel to the liver. And then the second last point, the parasite produced in the liver then re-enter the bloodstream, and then the parasite produce male and female gametes in the bloodstream of a person. So this is the life cycle of the malaria, or of this parasite which is called Plasmodium. So now, okay, let us move to our questions. Stage 2 host needed by parasites to complete its life cycle. We know that the 2 hosts are mosquito and humans. And then the next question is like this, okay, explain why a disease like malaria has an influence on the economy of the country? So this is how you have to answer this question. It is because it says people cannot go to work. So it means that if a person gets infected with malaria, so it means that they cannot go to work because of what? Because they will be having these symptoms, because of fever, vomiting, and headaches, and then which will lead to a loss of income. So that is how malaria affects the economy of the country. And then the next question suggests 3 control measures that can prevent people being bitten by mosquitoes and contracting malaria. So how can we manage this or control this? So it means that we will only have to use the insect repellent and then anti-malarial drugs and insecticides. So that is how we can manage or prevent ourselves from being infected with malaria. Okay. And then now we move to group number 4. Group number 4, which is kingdom fungi. Okay, and then let us look at examples of fungi. We have mushrooms, and then we also have yeast, and then ringworm, and then molds found on bread. Molds found on bread or cakes or fruits. And then these are all the examples of fungi. And then another thing that you have to know, fungi are not similar in appearance. They are totally different from the appearance. So one example of the fungi, it is the bread mold. And then we also have the mushroom, and then penicillium, and the last one, it is the ringworm. So ringworm, it looks like this. So let us look at the general characteristics of fungi. They are made up of eukaryotic cells. So it means that they are eukaryotes. So it also means that they have a nucleus. They are mostly multicellular, and then cells are arranged in a thread-like hyphae, and then hyphae, it is from mycelium, and then the cell wall contains chitin, and then they can reproduce asexually and sexually. So those are the characteristics of fungi. So we have fungi such as bread mold. Okay, I have mentioned the bread mold. Yeah. They produce their spores in special spore cases called sporangia. And then let us look at the effects and management of a ringworm. And so a ringworm, we are going to take a ringworm as an example of fungi, or one of the diseases which are caused by fungi. So this ringworm, it is caused by a fungus and not a worm. So when we go back to the diagrams of the fungi, so this is how the ringworm looks like. Here is the structure of a ringworm. So we will make use of these notes. So ringworm, let us look at the symptoms. They start as flat, scaly spots, which then develops a raised border that advances outward in a cycle. Most of the time, they are like this. So how can we manage these ringworms? By application of an antifungal infection. And then another thing that we can do, we have to wash our hands after handling animals, soil, and plants. And then another thing that we have to do, avoid touching characteristic lesions on other people. And then wear loose-fitting clothing. And then another one, it will be the practice of good hygiene when participating in sports, which involves physical contact with other people. So this is how we can manage or control or prevent ourselves from being infected with ringworms. So that was the last microorganisms that we were dealing with for today. And then before we move to another concept, let us look at our question and answer document that we have. And then on page 10 and 11, there is a question there on page 10 and 11. Okay. So this question is based on one of the examples of fungi, which is bread mold. So it goes like this. Question 2.2, a grade 11 learner wanted to investigate what factors help mold or fungi to grow on a bread. So she followed the following procedure. She took 4 slices of white bread and put different substances on each slice of bread as follows. So on slice number 1, it was left dry. Slice number 2, 20 milliliter of tap water, and then slice number 3, so this learner only put 20 milliliter again of lemon juice, and then slice number 4, it was sugar water. So step number 2, each slice of bread was put in a plastic bag and kept in a dark cupboard at a temperature of about 25 degrees Celsius for 2 weeks. And then step number 3, she observed the growth of mold on the 4 slices of bread and calculated the percentage area of the bread covered by a mold. Her results are shown in the table below. So this is the table of the results. And then first column, we have slice number 1 up to 4. And then second column, mold present on bread in percentages. And then let us try to interpret or analyze this table. So on slide number 1, the percentage of mold that grew, it is 0%. And then slides number 2, after 2 weeks, percentage of mold, it was 2%. And then slice number 3, it was 1%. And then on slice number 4, it was 4%, the one which has sugar water. So our first question is like this. Name the dependent variable of a investigation. So what will be the dependent variable? Let us look at the headings of our tables here. We have the slice and the mold present on a bread. So, and then this is our investigation statement again. This is where you can also find the dependent and independent variables. Grade 11 learners wanted to investigate, so they wanted to investigate what factors help mold or fungi to grow on a bread. So it means that our dependent variable, it will be percentage mold grown on bread. It depends on what? It depends on the slice of bread or the type of bread slice. So our answer, it will be percentage mold grown on bread. And then give the ratio of the percentage growth of mold on slice number 2 compared to slice number 4. Here is slice number 2, and then the percentage of mold is 2%. We will have something like this. It will be 2%, and then we compare it with slice number 4, which is 4%. So we have 2:4, and then we can see that we can also simplify this. So we will have, it will be 1 out of 2. 2 goes into 2, how many times? It is 1, and then into 4 is 2. So our answer will be 1:2. Explain why she included dry bread in her investigation. To act as a control or to compare results. Give 3 factors that must be controlled in this investigation to make this a valid test. So you remember that the controlled variables, and then validity. And then sometimes a question can be asked like this. What is it that we have to make to make sure that the results of this investigation are valid? Or how can we improve the validity of the results of our investigation? So the controlled variables, slices must be from the same bread. If we are using Sasco bread or Albany bread, so it means that slice number 1, 2, 3, 4, they will have to be from the same type of bread. So same bread. And then another one, the slice must be the same size. Same size of slices. And then another one, slices must be exposed to the same light intensity, temperature, or environmental conditions. Same environmental conditions. So those are the control variables or factors that can be kept constant, or factors that can increase the validity of the test or the investigation. So suggest why mold did not grow well on the bread sprinkled with lemon juice. When you look back on our table, bread number 3, the one sprinkled with lemon juice, And then the results that we got here, it is 1% growth. So you have to suggest a reason why the mold did not grow well on this bread. It is because lemon juice is acidic. So we know that acids, they inhibit the growth of mold or fungi. It is acidic, and then acids inhibit the growth of mold or fungi. All right. So that was one of our questions, which was based on one of the examples of fungi, which is bread mold. All right, and then now we move to the second part of our lesson. So it means that we are done with this part. Basic structure, the one of basic structures and characteristics, together with the one. We have also covered this part of the effect and management of diseases of each of the microorganisms. So now let us move to the roles that these groups, which groups? The groups that we have mentioned, virus, bacteria, protists, and fungi, the role that they play in maintaining the balance in the environment and the web of life. And then we will also combine this with symbiotic relationships of bacteria, such as this is where we will deal with nitrogen-fixing bacteria and E. coli in the human intestines. So, role of micro in maintaining balance in the environment and the web of life. So sometimes you will get a question which will be like this during the examination. Explain or briefly discuss or describe the role of microorganisms in maintaining balance in the environment and the web of life. So this is how you have to answer this question. You remember that some of the microorganisms, like as we have said, that they are autotrophic. So autotrophs or autotrophic organisms, you still remember that they are able to manufacture their own food. By making use of light energy or chemical energy. And then after that, to make food which carries the energy to the consumers, so which are heterotrophs. So heterotrophs So we have said that heterotrophic organisms, they are unable to manufacture their own food. So autotrophs, they manufacture their own food, and then by making use of, by the process of photosynthesis. And then after that, they carry this food to where? To the heterotrophic or heterotrophs organisms, which are consumers. And then you still remember that, let me show you this flow diagram, which will explain this. You still remember that we have small organisms which are found in water, and then we call them the planktons. So these small organisms, they will be eaten by the small crustaceans. And then after that, energy, it means that energy will be transferred from one organism to another. And then the small organisms, they will be crustaceans, they will be eaten by the sardines. and then sardines will be eaten by whales, humans, and sharks. So this simply means that the energy trapped in the food produced by the planktons. You will remember that planktons are those small organisms or autotrophs which are found in water. So they produce their own food, and then after that there is that energy which is trapped in the food that have been produced by what? By the planktons. So this energy will be transferred from one organism to another. So this is what happened here. The energy will be transferred to these organisms, and then these organisms, and then sardines will be eaten by whales, humans, and sharks. So it means that energy will be transferred from sardines to all these organisms that you see here. And then we call all this process, we call it the energy flow or web of life. And then after that, another thing that will happen, that we'll have to mention is that autotrophic organisms like this one that we have mentioned, they use up carbon dioxide. The planktons, we know that these are the small plants which are found inside water. So they will use up carbon dioxide and release oxygen. They release oxygen and then they use carbon dioxide, and then during the process of what? Of photosynthesis. So in this way, there will be a balance between the oxygen and carbon dioxide. So it simply means that when we go back to point number 2, you will have to remember that autotrophs or autotrophic organisms, which are plants, small plants which are found in water and then which are called the planktons. They use what? During the process of photosynthesis, they use carbon dioxide, and then after that they release oxygen. And then in that way, so there will be that balance between oxygen and carbon dioxide. And then point number 3 that you will also have to mention is that some of the microorganisms, they act as decomposers. And then they break down organisms that have died. So it means that dead organic material will be broken down by what? By these microorganisms that we call them the decomposers. As well as their waste products. So breaking down of organisms that have died as well as their waste product. And then this is what is going to happen. During that breakdown of organic material within the dead organisms, so it means that this organic material or dead organic material which will be broken down into water, carbon dioxide, ammonia containing nitrogen, and heat energy. And then all these substances will be recycled or returned back to where? To the atmosphere. And then another thing, which is the last point, some of the microorganisms are involved in symbiotic relationships such as mutualism, parasitism, and commensalism. So please remember that things that you will have to say when you answer this question, like as I have said, that sometimes they will ask you to briefly describe the role of microorganisms in maintaining the balance in the environment and the web of life. So bullet number 1, it will be this one of organisms that live in water, small plants that live in water, and then they will be, energy will be transferred here from one organism to another. So these small plants will be eaten by the small crustaceans, and then by sardines, and then sardines will be eaten by all these organisms, which are whales, humans, sharks, and other organisms which have not been mentioned here. So, like as I have said, that the energy which is trapped from the food produced by the planktons, then it will be transferred to all these organisms. And then another thing, these organisms also, again, the autotrophic organisms, during the process of photosynthesis, they use carbon dioxide and then they release oxygen. And then by so doing, it means that they will be able to maintain the balance between oxygen and carbon dioxide. And then another point, it is this one of decomposers. So some of the microorganisms which are decomposers, or they act as decomposers in breaking down organisms that have died as well as their waste products. And then during the breaking down of these organisms, this is what will happen. Substances such as water, carbon dioxide, ammonia containing nitrogen, and heat energy will be released in the atmosphere or back in into the atmosphere or the environment. So it means that they will be recycled. And then the last thing, it is this one, that some of the microorganisms or other microorganisms, they will be involved in symbiotic relationships such as mutualism, parasitism, and commensalism. So let us look at symbiotic relationships. What does symbiosis mean? It refers to an interaction into two organisms of different species that live together in a direct contact. So we have the three types of interactions, and then the first one is mutualism. Mutualism, this is where both organisms in the relationship benefit. Both organisms in the relationship benefit. And then the second type of these relationships, it is parasitism. One organism benefits, one benefit, and the other one is harmed. So it means that it doesn't benefit and it is also harmed. And then the third one, it is commensalism. So commensalism, one organism benefits by using the other only for support. So it simply means that the other organism or the second organism, it is not harmed. It doesn't benefit, and then it will only be used for support, and then again it will not be harmed. So let us look at one of the examples of mutualism. So we have, we are going to do two, which is the nitrogen fixing bacteria and the E. coli in the human intestines. So one of the examples of mutualism is nitrogen fixing bacteria. So you remember that plants and animals, they require nitrogen. Plants and animals, they require nitrogen for what? nitrogen for the formation of proteins. All right. So plants and animals, we require nitrogen for the formation of proteins, and then 79% of the air in the atmosphere is nitrogen. So we have nitrogen, which is 79%. So 79% of the air in the atmosphere is nitrogen. But we know that plants and animals, they are unable to absorb nitrogen directly from the what? From the atmosphere. So it means that this nitrogen, it will have to be converted into nitrates. So who will convert this nitrogen in the nitrates? It is this bacteria, which is called the nitrogen fixing bacteria. So nitrogen fixing bacteria, it will fix or convert nitrogen from the atmosphere into nitrates. Because of what? Because plants and animals are unable to absorb nitrogen directly from the atmosphere. And then after that, this is what is going to happen. Now plants will be able to absorb nitrogen, but in a form of nitrates from the atmosphere. So plants now will absorb nitrogen, but in a form of nitrates. And then what about the animals? So it means that animals will now get what? Nitrogen in a form of nitrates from the plants that they eat. So animals will get this nitrogen from the plants. So that is all about the nitrogen fixing bacteria. And then the last thing here on the nitrogen fixing bacteria. So some of these nitrogen fixing bacteria, we know that nitrogen fixing bacteria, they are found all over in the atmosphere, and then but some, they live in the root nodules of plant. So in return, this is what is going to happen. The nodule bacteria, so now we call them the nodule bacteria, the ones that live in the root nodules of the plants. So nodule bacteria, they will obtain carbohydrates from the plants. And then what will happen again? So the plant benefits by getting nitrates from the bacteria, whilst the bacteria benefit by getting carbohydrates from the plants. So this is an example of mutualism. So I'm just going to make a summary of this or repeat this again. So please remember that we said that nitrogen fixing bacteria in plants and animals. So it is an example, nitrogen fixing bacteria in plants and equally in the human intestines. They both show the type of symbiosis, which is mutualism. So when we start with the nitrogen fixing bacteria, we said that we know that plants and animals, they require nitrogen for the formation of proteins. And then 79% of the air in the atmosphere, it is nitrogen. But now the problem is that animals and plants, they cannot absorb nitrogen directly from the atmosphere as is. So it has to be converted into nitrates. So we have this bacteria which is called nitrogen fixing bacteria. So it will fix this nitrogen, or it will convert it from nitrogen, it will convert it back into nitrates. And then in this way, plants will be able to absorb nitrogen in a form of nitrates. And then after that, animals will also get nitrogen from the plants in a form of nitrates when they eat these plants. And then after that, some of these nitrogen fixing bacteria, they are found in the root nodules of the plants, and then we call them the nodule bacteria. So they benefit from the plants by getting carbohydrates. And then plants also benefit from this bacteria by what? By what? We have mentioned that the bacteria is responsible for fixing the nitrogen into nitrates. So that is why we say that the type of relationship that takes place here, it is mutualism, where both organisms benefit. The bacteria benefits, and then again the plants also benefit. So let us look at E. coli in the human intestines. This is another example of mutualism. So E. coli in the human intestines. We know that we have different species of bacteria in the human intestines. But these bacteria, they are harmless. Different species of bacteria which are harmless in the human intestines. And then there are about, it is estimated that they are about 500 to 1,000. So these bacteria, they are mutualistic bacteria. So how are they benefiting from the human intestines? They benefit by obtaining the food. So how are human beings benefiting from this bacteria? So number one, we benefit in this way. So point number one, it will be this bacteria, they help decay indigestible food into feces. And then point number two, they synthesize, or they make vitamin B, E, and K for our use. So the mutual relationship that takes place between the bacteria that are found in the human intestines together with us as human beings, it is as follows. The bacteria, they benefit from us by getting food from our small intestines, and then we also benefit from the bacteria in such that they help decay indigestible food into feces, and then again they synthesize, or they make vitamin B, E, and K for our use. So let us move back to that document which has questions. I have one question here which is based on this concept. So this was one of the questions which was asked, I'm sure, in 2017 or 18. So our question is like this: explain why the relationship between E. coli and humans can be seen as mutualism. So this is how this question was answered. Both organisms benefit, and then it was 1 mark, which is correct, from the living relationship. From this relationship, they both benefit. And then the bacteria live in a protected environment and are provided with good supply of food. You will remember that I have also mentioned that these bacteria, they benefit from us by getting food, or they benefit from our small intestines by getting food, and then allows bacteria to grow and safely reproduce in large numbers. And then after that, if they get food, and then they will reproduce and grow in large numbers. But we still remember that these bacteria, they are harmless. So the bacteria provide the human body with extra vitamin K and helps with the absorption of digested nutrients. So this is how we benefit from this bacteria. So they provide the human body with extra vitamin K, and then I have also said also the vitamin E and B. All right, so that was our question, which was based on that type of relationship, which is mutualism. So now we move back to week 3 of our ATP, which is immunity. So immunity, we have 2 types of immunity, and then the first one is passive immunity, and then the second one is active immunity. So what is passive immunity? It is the mechanism to prevent pathogens from entering. They prevent You still remember that I have said that pathogens are disease-causing organisms. So passive, it has to do with what? They prevent pathogens from entering, or disease-causing organisms from entering. And then active immunity. It is a response mechanism. So active immunity, it has to do with this. The response mechanism to fight off infection once the pathogens or the disease-causing organisms have already entered into the human body. So that is the active immunity. So let us look at passive immunity. This one where we said that it is the response mechanism that prevents the pathogens or these microorganisms or disease-causing organisms from entering our bodies. So passive immunity mechanism used by plants and animals. And then let us look at, we will start first with animals. Disease-causing microorganisms or pathogens are prevented from entering our bodies in the following ways. So, this is what we have. So, how are these disease-causing microorganisms or pathogens prevented from entering our bodies. Then the first one, it is mucus in the nose and throat. So that mucus in the nose and throat, just know that it prevents the microorganisms or pathogens from entering into our bodies. And then cilia lining in the nasal passage and the trachea. And then tears in the conjunctiva of the eye. And then hydrochloric acid in the stomach. It also prevents the microorganisms or pathogens from entering in our bodies. And then unbroken skin. So it means that broken skin, it will allow the pathogens to enter into our bodies. But this is based on what? On animals. But then when we move to plants, this is what happens. The method that can be used to prevent infection by the microorganisms. And then number one, it is this one. A thick bark of woody plants protect the more delicate tissues inside. So the second one, the waxy cuticle holds and moisture in moisture and keeps out microorganisms. And then sticky secretions given off by plants. It makes difficult for the insects and animals to eat that plant. And then some of the chemical secretions of some plants are poisonous to many organisms. So that is all with passive immunity in animals and plants. So now let us move to active immunity. And then you still remember that I have said that active immunity, it is the response mechanism to fight of infection once the pathogens have entered. So with this one, we don't prevent the pathogens from entering. Already the body is infected. So what will be the immune response mechanism? So this is what happens. When these disease-causing microorganisms or pathogens or microbes, when they enter our bodies, so this is what will happen. The white blood corpuscles in our blood in the blood will produce the antibodies. So it means that when these organisms, once they enter into our bodies and pass through some of the defenses, and then what is going to happen, the white blood cells or corpuscles, they will produce what we call antibodies. And then each antibody can only fight a particular microbe or microorganism. So we say that the antibodies, they are specific to the microbe they attack. So each and every antibody produced will only be able to fight a particular microbe. So they are very specific to the microbe they attack. But some of the antibodies might be able to fight closely related microbes, such as what? Such as, for examples, antibodies produced for disease such as cowpox can also fight off smallpox. So this means that although we know that antibodies which will be produced once the microbes enters in the human body, so we have said that they are very specific to the microbe they attack, but some of them we know that they can also fight closely related diseases such as cowpox and smallpox. So it means that maybe an antibody which is being produced for cowpox, then it will also be able to fight off smallpox. So this ability of the body to produce antibodies is called immunity. So please, you have to be able to describe this term. So immunity, it is the ability of the body to produce antibodies. So we have again what is called, we started with the passive immunity and active immunity, and then again we have artificial immunity and natural immunity. And then let us look at this document that I have here, and then we will go through this part. So, and then, like as I have said, that immunity, it is the body's ability to produce antibodies. Then I'm just going to read here. Almost all of us have an inborn ability to produce these antibodies, and we call this inborn ability natural immunity. So it means that all the people, all the human beings, they have what is called the natural immunity. We can also obtain or acquire. So now we have the natural together with acquired immunity. The natural immunity, it is the one that we are born with, and then after that we have acquired immunity. This one, we acquire it during our lifetime, and then this is how we get it. We get acquired immunity. And then point number 1, a person gets a mild form of a disease through natural infection. So it means that you will be infected, maybe by a certain or specific disease. And then after that, once you get infected, so it means that the microorganisms or microbes, they have entered into your system. And then what is going to happen? The body will start to produce the antibodies to fight that disease. And then after that, you will get better, and then the antibodies remain in your body. So it means that after the antibodies, like as I have said, that your body will be able to produce the antibodies, and then after that, you will start getting better. So after that, it will mean that those antibodies, now they will remain in your system. So that is what is meant here. And then these antibodies fight off any further infection of the same disease. So, and then with that, it will mean that you have acquired what? Immunity. So remember that the difference between natural immunity and acquired, natural, we are born with it. And then acquired, and then this is how you get acquired. You will start getting sick first, or you will be infected again by any type of disease or microbe. And then once you get infected, what will happen? Your body will produce the antibodies to fight that disease, and then you will get better. And then after getting better, so it will now mean that you will now have the antibodies. And then those antibodies, then they will continue fighting off any further infection of the same disease. So with that, let us move to page 8 to 9 before we move to vaccinations. Page 8 to 9 of our, of our question and answer. So this is what we have here. So it was question 3. The flow diagram below shows 2 methods which are used to give humans immunity against diseases. Method number A shows active immunity, and method number B, it shows passive immunity. So you remember that we said that the difference between active and passive immunity. So active, all right, active, it is a mechanism to prevent pathogens from entering. So with this one, we are preventing these microbes or microorganisms from entering into our bodies. And then passive immunity, this one already the microbes or microorganisms have entered into the body. So that is the immune response mechanism to fight off the infection. So it is after the pathogens have entered. So now we will have that immune response mechanism. So that is method number B. Method number B, I will just write here, it is passive, this one. And then method number A, they have said that it is active. All right, so we have our diagram here in a form of a flow chart. Okay, so method used to give humans protection, and then we have method number A and method number B. And then let us start with method number A, which is active immunity. Step number 1, a person receives small dose of rabies virus. So they have made use of what? Of rabies virus in this example, and then which is an antigen. So that small dose, we call it an antigen. All right. And then you still have to remember that with this method of active, it means that we are preventing the pathogens from entering. The person is not yet being infected with what? The microorganism or rabies. So this person will receive a small dose of rabies virus or antigen, and then what is going to happen? The virus will multiply slowly in the body of this person. And then what will happen when this virus multiplies? And then it will be harmless. So the antibodies will be produced. So that is why you see antibodies are made. And then after that, the person will receive small dose of rabies virus antigen again. So that is active immunity. But now with method number B, already this person is being infected or has been infected with the microbes or pathogens. So this is what happens. Tuberculosis, so they have made use of TB in this example, or antigen TB injected into an animal, and then antibodies are made within the animal's body. So the blood removed from the animal used to produce a serum with antibodies, and then this serum will be injected into a person infected with tuberculosis, and then tuberculosis antibodies are weakened in the person's body. So that is method number B of passive immunity. So let us look at the questions. So here are the questions. Name the substances that are produced by the body which destroy harmful viruses and bacteria. And then, like as we have said, that we call them antibodies. What does method A give? Why does method A give long-lasting protection against rabies? And then it is this method of active immunity. So it is because the antibodies, they will remain in the body, and then the body will continue to make antibodies. So that is why method A gives a long-lasting protection against rabies. So question 3.1.3, why does method B not give long-lasting protection against TB, which is tuberculosis? And then it is because antibodies are weakened after a short time, and then the person has not made his or her own antibodies. antibodies. (3.1.4) In immunization against rabies, a second dose of the weakened virus is given. This is known as a booster. Suggest why this booster is necessary. You remember that, okay, with rabies, we had a small dose here. And then again, here is the booster, another dose. Why is this necessary? It is because so that more antibodies are made, we give the first antigen, and then after that, the second one, which is called the booster, to make sure that more antibodies will be made, to keep the antibody count high, or to keep the antibody count high, so that the body keeps making antibodies for a long time. So the last question, question 3.1.5, why is method B very good for delaying quickly with an infection like tuberculosis? Okay, sorry, that is for, let me read this question again. Why is method B very good for dealing quickly with an infection like TB or tuberculosis? So the body does not have to wait for antibody formation, and then antibodies start working straight away with method number B. All right. And then let us look at vaccinations. So you remember that we talked about natural immunity and acquired immunity. Acquired immunity, the one that we talked about, it is that one where a person will have to be to get infected first, then the his or her body will start producing the antibodies. And then another form of immunity, it is vaccinations. So vaccinations, it is another type of acquired immunity, and then it is obtained through artificial means, such as by a vaccine. It is artificial and in a form of a vaccine. All right, and then it means that you receive, a person who is vaccinated, we have to know that they receive a weakened form of a germ, either by injection or swallowing a tablet or a syrup. And then the body of a person who is vaccinated will produce antibodies. So it means that once you start, or after you get, after getting a vaccine, this is what will happen. Your body will start producing the antibodies. So your body will start producing the antibodies to fight off the infection. And then since vaccines contain a weakened form of the germ, or a weak form of the microbe, and then a healthy person who is vaccinated will not get this disease. And then antibodies produced within the body will now protect him or him against the new infection. So in other words, that person, it will mean that they have acquired immunity through vaccination. So by a means of vaccine, and then is immunity through vaccination. So in case of the plants, so this is vaccination based on animals. But now in case of the plants, When part of a plant becomes infected with a bacteria or a viral pathogen, so this is what happens. That part which is infected, it will quickly die off and shed off to prevent the spread of the disease to other parts of the plant. And then this is known as, we call this localized hypersensitive response, and then which is called HR. Then the plant uses what is known as systemic acquired resistance. So in an attempt to make the entire plant resistant. So this is how plants get an artificial immunity. All right. And then there is another part from this document that I have, and I'm sure is the last part on this thing of immunity. So I will just go through the whole part. So I have also indicated that you have to read this part. And then let us check this, so that for you to be able to understand mutations, then we will have to start here first. When a disease, they say early response of the disease-causing microorganisms on entering the host. When a disease-causing microorganism enters its host, one of the first things it does is to reproduce itself very rapidly. And then let us say, for example, let me take the COVID-19 virus now. So maybe we can use that as an example here. And then the first thing that it does after it has entered the human body, it reproduces itself very rapidly. And then, okay, here they made use of the HIV or the HI virus, the one that we used to call HIV. For example, when it enters the human body, it causes human cells to make many copies of the HI virus. So you see this HI virus, once it enters into the body, and then it uses the what? The human cells to help it to make copies of itself. And then after that, when the malarian, okay, we have another example here, the malaria parasite. You remember that we have said that it is caused by a parasite or microbe called Plasmodium. It first enters a human host, and then after that it will quickly reproduce to produce about 24 copies of itself. And then within a duration of what? Of 10 to 14 days, it will reproduce into 24,000 or more copies of itself, and then within 10 to 14 days. So similar patterns of very rapid reproduction occurs in the TB bacteria, even also with the bacteria which causes the TB. Once it enters into the human body, and then it quickly reproduces to make as many as possible, as many copies as possible. And then the purpose of this rapid reproduction is to make a large number of copies of itself, is to make certain that at least a few of the parasites survive, even if one or two die because of the host natural immunity. So why are these microbes reproducing so rapidly? So they just want to make sure that they make large number of copies of themselves to make sure that as soon as the body responds via natural immunity, so at least one or even two microbes will survive. So not all the new copies of the microorganisms are identical. So now after they have reproduced themselves, you will find that the new copies are no more identical, or not all of them will be identical to the original ones. So let me check here. They say not all the microorganisms are identical to the virus bacteria. Okay, they give examples here. protists or fungus from which they were originally produced. So some are slightly different. These new or slightly different copies are now called mutations. So you have to understand that, like as we have said, that once the microbes enters into to the human body, they will start reproducing very rapidly, and then after that, to make as many copies as they can. And then after that, out of all those copies that will be made, some of them, they won't be identical to the original strain. So we will call that mutations. So it means that they have now caused mutations. So let us look at the effect of the use of the drugs on microorganisms. So now you will be able to understand the term mutations because it will be mentioned on this part. So the effect of the use of drugs on microorganisms. Again, I will go through this extract. So when a patient takes medicines such as drugs, the drugs kills the pathogens. So we know that once you are being infected with these pathogens, and then you will start taking medication or drugs. And then after that, you will get better, like as it has been mentioned here. And then in some cases, an infected person is completely cured. And then in some other cases, and then after taking the drugs or medication or antibiotics, we know that you will be completely cured. But in other cases, such as HIV, look at this one, HIV/AIDS, you won't be completely cured. But this is what is going to happen. All the drugs that are available at the present time can only slow down the disease. The disease will only slow down from becoming worse. So that is, the drugs cannot cure the infected person of the disease. So we know that the drugs that are being used now for HIV/AIDS are the ARVs. So it means that the ARVs, they cannot completely cure HIV/AIDS, but the only thing that they do, they slow down the disease from becoming worse. So that is all that has been described from this extract. And then another thing, in some cases the drugs work at first and then become ineffective. So you will find that sometimes these drugs that are used to cure or to slow down the disease which are caused by the microbes, and then they will start to work at first, and then after that they will become ineffective. So it is because the drugs are able to kill the original strain, and then it is because of what? If they start working at first and then after that they become ineffective, it means that they were able to kill the original strain of the disease-causing microorganisms. But now they are unable to kill the second strain, and then this one that we call it the mutant strain or mutant copies. So this takes us back to where? To this paragraph or extract that I started with, the one that we read. So, and then because of that, then we will say that the microorganism has developed immunity or resistance to the drug. So there are reasons why this mutation sometimes happens, or the resistance to microorganisms, resistance of microorganisms to drugs happens. It is because of the following reasons. Sometimes the drugs themselves can cause mutations. Some of the drugs, they can cause mutations. So you will start using them, and then they will work, and then after a certain period, they will start to be ineffective. And then sometimes you will find that the main reason is with what? With the drugs themselves. So the drugs then don't work on the mutant organism. So stopping the drug treatment, even if it is temporarily for a few days, sometimes it can cause the resistance in the microorganisms. So this simply means that, let us say, you become infected with one of the microorganisms, and then you consult a doctor, and then they prescribe the antibiotics. Then you start using them for day 1, 2, 3, maybe the course of the antibiotics, and then you've been told that it is 7 or 10 days. So you use them on day 1, day 2, day 3, and then you skip day number 4. You forget to take them, and then you continue with day number 5, and then that thing, it might lead to what? To drug resistance, and then you will find that the antibiotics, they become ineffective. And then another thing which can cause resistance of microorganisms to drugs, it is this one of the treatment with combination of drugs. Sometimes they call it cocktail of drugs, reduces or prevents the microorganisms from becoming drug resistant. So that is all with what the effect of use of drugs of microorganisms. So the last two things are these ones from our ATP, the use of microorganisms to produce medicine and traditional technology. So use of microorganisms to produce medicine, and then many bacteria and fungi are used to produce vitamins. They are used to produce vitamins, proteins, hormones, and antibiotics. And then again, they are used in the production of penicillin, which is an antibiotic. And then the last part, it is this one of the traditional technology to produce beer, wine, and cheese. And then you know yeast. And then yeast, which is commonly used for baking. It is an example of a fungi or fungus. So these yeast cells, they will undergo a process called anaerobic respiration, and then you will deal with anaerobic respiration later this year. And then this is what happens during anaerobic respiration. The yeast cells, they convert the sugar into alcohol and carbon dioxide. So the carbon dioxide produced in this way, it will be used to cause the dough to rise during baking. And then after that, this is how this thing of the traditional technology to produce, okay. So with this one, it is the production of what? Of bread using the yeast cells. So it means that if you are, before you bake, let me say for an example, you want to bake bread by using the yeast cells. So you will buy yeast, and then after that you will mix it with the flour or whatever, and then after that anaerobic respiration will take place, and then after that the dough will rise. So when it rises, it means that these yeast cells, they are now converting what? The sugars to alcohol and carbon dioxide. So that is why you will see it rising. And then another last thing, some of the bacteria called lactic acid bacteria, we have bacteria that are called lactic acid bacteria. So they produce lactic acid through anaerobic respiration again. And then the lactic acid reduces the pH of the milk and makes it to taste sour. So humans have made use of this bacteria in production of cheese and yogurt production. So it means that there is this bacteria which is called the lactic acid bacteria, and then it is being used in the production of what? Of cheese and yogurt. So it produces what is called the lactic acid, and then by the process of anaerobic respiration. So what happens here? The lactic acid, this lactic acid, which is being produced by lactic acid bacteria, it reduces the pH of the milk, and then it will start to make this milk to taste sour, and then after that human will use that to produce cheese and yogurt. So grade 11 learners, this brings us to the end of our lesson today, and then it was based on week 1 to week 3 topics, mainly which was biodiversity and classification of microorganisms. So for me, and then I just want to say thank you so much for watching this video or lesson. Goodbye.